Greetings for the day. This is Vikrant Arya and I am here to deliver a talk on parameters for herbal drug standardization. So this lecture was delivered at RIISM Joginder Nagar and special thanks to Dr. Pankaj Palsra, Principal Investigator RIISM Joginder Nagar, who gave me this opportunity to deliver my talk there. Yes, first of all, I will acknowledge all the sources that I will be using to prepare this lecture, like Ayurvedic Pharmacopy of India, WHO guidelines, and other parameters like Protocol for Testing Ayurveda, Siddha, and Unani medicines. I have obtained all the information from the reference sources only. Let's start the topic. After watching this video lecture, you will be able to know about the herbal medicine basics, then there are different types of standardization parameters, and a brief about the procedure also that are prescribed by different authorized databases or sources. And of course, there are different types of herbal drug evaluation parameters that we will cover in this video lecture. So first of all, there are different terms related to the herbal medicine, the herbs or the herbal preparation. So as per WHO guidelines on good agriculture and collection practices for medicinal plants, herb may be defined as any plant material such as leaf, flower, fruit, stem, wood, bark, etc., which may be entire, fragmented, or in the powdered form. So there is a simple definition of herb as per WHO. Then next definition of herbal medicine as per WHO again. This includes the herb, herbal materials, herbal preparation, and also finished herbal products. Next one is the herbal preparation. So basically, herbal preparation nothing but these are the you can say the finished herbal products and may include the committed or you can say the powder form, herbal materials, herbal extract tinctures or other herbal materials. So they are also obtained by the process of some extraction, fractionation, purification or even the concentration or other physical or biological processes. And they also include the preparation made by steeping or heating herbal preparation in the alcoholic beverages or honey or in other materials. So about the standardization. So the term standardization simply means it is a process or you can say the standard set of procedures which help us to determine the identity, quality, purity, safety and also the efficacy of the drug. Yes, there are different standardization parameters that we covered also like organoleptic, botanical, physical, chemical and the biological. We will cover them each and every aspect one by one in a brief. So first one is the organoleptic. As we know, these are the morphological parameters like color, odor, taste, size, shape, texture and of course the fracture. So by observing with the naked eye or using in the magnifying glass simply we can able to predict about the morphological characteristics of different herbal medicine or crude drugs also. So these are the organoleptic parameters or you can say the organoleptic evaluation. So this is the basically the organoleptic characteristics and in the allopathy system of medicines we are generally using some parameters for the color also. So this is the Munsell suggested by the Munsell, that is the color system that is known as the Munsell system suggested in 1913 by an American art instructor and the painter, and we are still using in the allopathic system of medicine but it lacks in the Ayurveda system of medicine. So we must have some standard parameters for the color also because this is also very important parameter. But I cannot say that I'm not saying that there are not any standard parameter, but it should be synchronized in such a way that it should be documented all over and people can use that also. So there are different examples that I mentioned that I found from different sources. So there are examples of some drugs like Brahmi which are grayish green in color and odor is characteristic, taste is bitter. You can see the size also 7.5 to 115 centimeter and width is 1.5 to 6.5 in diameter and the shape is entire crenate. This is the leaf shape because the part used of Brahmi's leaf also the herb is brain tonic. Next is the cascara bark. Here is the bark. The bark size is the of course in the form of quills or channels. Here is the examples of different types of shapes of the leaf like linear, oblong, lanceolate, crenate, or you can say the ovate, ovovate, and these are the shapes of the different bark like flat, curved, recurved, channelized, quill, double quill, etc. So next one. Yes, there are the different morphological characteristics like sarpgandha, that is just look like serpent-like shape, sarpagandha. Another plant, Paris ginseng, it has a shape of human body. So it is widely used as a also as an adaptogenic immunomodulatory drug, and it's a drug of also of the Chinese system of medicine. The next one is the botanical. As we know, after morphological, after viewing the external characteristics, we have to go for the internal visualization using some devices like microscope is the most commonly used device or the instrument that we are using for botanical examination. And in botanical examination, we know, yes, the internal structure that we'll be able to see with the help of the microscope. So this method allows a more detailed examination of a drug, and it can be used to identify the organized drug by their histological character. So we have to focus upon the plant tissues like parenchyma, collenchyma, sclerenchyma, etc., etc. So, and we also know that different plants have different characteristics. If we talk about the leaf, so in the tissue of the leaf, it had different characteristics, whereas in the root, it has different characteristics, and seed, stem, bark has also its particular characteristic. That is, we are talking about the microscopic characteristic of a particular plant part. So yes, there are different botanical parameters that we also determine in the standardization parameters. Like if we talk about the leaf, then we calculate stomatal number, stomatal index is there, weed islet number is there, weed termination, and the palisade ratio, of course. So we will go the definition again of each one by one. First of all, the definition of stomatal number. So stomatal number, it is defined as the average number of stomata present per square mm of the epidermis of the leaf. Simply, very simple. Second is the index that we are calculating after calculating the stomatal number, of course. So there is a formula of calculating the stomatal index, that is SI, stomatal index is equal to S divided E plus S into 100, which here means E is the epidermal cell, and whereas the S is the number of stomata per unit area. So yes, there is a device or you can the instrument that we are using to calculate the stomatal index or the weed islet number, weed termination number is the camera lucida. So this is the camera lucida, and it has a detailed procedure that with the help of this camera lucida, it will attach to the microscope that we are using in the laboratory like compound microscope, and we can easily calculate the stomatal number, stomatal index, even the weed islet, weed termination, and the palisade ratio also. So about the definition of weed islet number, it is defined as the number of weeds islets that are present per square mm again on the leaf surface. And of course, midway between the midrib and the margin is the weed islet number. And second one is the weed termination, again the number of weed termination present per square mm midway between midrib and the margin. So it can calculate it by using camera lucida. And of course the palisade ratio is there. It is the number of palisade cell beneath each epidermal cell. So here is a diagrammatic representation also that we are calculating the stomatal number here, index, and of course the weed islet and termination number over here. Next one is the, again, yes, nowadays we are using electron microscopy and particularly scanning electron microscope, that is known as the SEM and TEM also, for the proper or you can say the deeper or more clearer identification of these plant tissues or the herbal drug or the crude drugs. So yes, these are the few images. As you can see, this is very precise and accurate images of the electron microscope that we are using for the botanical evaluation for herbal drugs also. Next, the determination of foreign matter. This is very simple parameter. In this, we are looking for the different organic content that are present, and it is separated by using simply by a magnifying glass or naked eye or unaided eye. So in this procedure, we generally weigh about 100 to 500 gram of the drug that we have to examine, and with the help of, you can say, the unaided eye, or even say simply the using a lens, magnifying glass lens, using a 6x, we can easily separate out the unwanted material that we are not required, or you can say the apart from the drug, other than the material, we easily separate out. Next one is the identification of moisture content. Again, very important parameter is there, and we all know there is a temperature requirement for calculating the moisture content, that is 105 degrees Celsius for 5 hours. And generally, because this procedure was generally adopted, done in a hot air oven, but nowadays we have different instruments, like as you can see in the image, this is the IR moisture balance. So in this instrument, we are easily able to determine the moisture content. So yet again, this is a physical parameter. After botanical, this is again the very important physical parameter for calculating for the herbal drug evaluation, crude drug evaluation. So after moisture content analysis or the moisture analysis, There is another very important parameter prescribed by different pharmacopoeias like Ayurvedic Pharmacopoeia of India, that is the determination of extractive value. And in API, alcohol soluble and water soluble extractive values are mentioned and compulsory or mandatory for calculating or maintaining the standardization parameters. So yes, we simply, for extraction purpose, we simply use the maceration procedure over here. So we simply do the maceration of particular quantity of the drug in a particular container, and for a period of certain hours we can do the maceration procedure. And after obtaining the extract, we can easily calculate the extractive value. So here is the procedure. We can simply take 5 gram of the drug as per this procedure and add in the 100 ml of alcohol, and over alcohol here means ethanol. We are generally using the ethanol, not the other alcohols like methanol or IPA. We are generally using the ethyl alcohol for a specific strength, and we will generally do the maceration for at least 24 hours. Now after shaking, in between we will do the shaking for at least 6 hours, and they'll allow to stand for 18 hours again, and then filter. After filtering, we will evaporate by using this the hot, or you can say the water bath, hot water bath, and we can easily obtain our dried extract, or you can say the semi-solid extract, and calculate the extractive value then again the same procedure was adopted for again water soluble. Over here, despite from alcohol, we are generally using water as a solvent. So this is the physical parameter again. Yes, very important. Ash value determination, of the inorganic impurities present in the plant sample. So this is the parameter for determination of inorganic impurities in any plant material. We will do the physical parameter of ash value. So what is ash? It is basically a residue remaining after the incineration of the drug, and it has a special procedure, and we are using, just in the case of, you can say, the moisture content analysis, we are using the hot air oven or IR moisture balance, and for the ash value we are using a special instrument or a device, that is the muffle furnace. So this is a muffle furnace. It has a special quality that it can, in this furnace we can increase the temperature up to 1000 degrees Celsius, and for the calculating of ash particularly we need 450 degrees Celsius. So we can do what generally we do. We can take a crucible, previously accurately weighed crucible was taken, drug was added in the crucible, and it was subjected to the incineration for a particular time period, that is in a particular temperature, that is 450 degrees Celsius, until it become a ash. How can we predict that the drug is converted to ash? When it has, when all the carbon content is removed, and we all know ash has a grayish color. So it will give an idea. I'm not saying that all the plant material will give a grayish color ash. It is not for all the crude drugs, but it has a general procedure that we can do, that is removal of the carbon, or you can say the black color impurities जब खत्म हो जाएंगी हमारी crucible में, then we can say that it might become the ash. So we will do the entire procedure. We have general procedure for different plants also. So next one is yes, ash value different types, like acid insoluble ash value, water soluble ash value, sulfated ash is also there, and these all determination of ash value will give us an idea about the impurities present in the samples, and we have standard procedures or standard ash value also, that is documented in the different pharmacopoeias like Ayurvedic Pharmacopoeia of India. Over here example is also given, like over here you can see the total ash value of amla. Example is amla as per API Ayurvedic Pharmacopoeia of India part 1 volume 1. Total ash not more than 7%. Suppose if your sample has more than 7% ash, what does it interfere? What does it indicate? It indicates that your sample is not of good quality. It might contain more amount of inorganic substances, and if you will prepare the formulation from that particular drug, it might cause some lethal or toxic effects also, because it has increase in the inorganic content and might cause decrease the pharmacological action of the drug and increase the toxic effect of the drug. So yes, next parameter is again very important, that we generally do for oils like volatile oils and fixed oil in case of herbal drugs. So this is parameter is known as the refractive index. Okay, refractive index is very simple parameter. As we know, there is a famous Snell's law, sin I upon sin R. अगर एक गिलास पे हम पानी का गिलास ले, उसमें एक पेंसिल रख दे या पेन रख दे, अगेन ऐसा लगता है कि वो पेंसिल दो हाफ में टूट गई है due to the refraction of the light. It's a simple parameter, sin I upon angle of incidence upon angle of refraction is there. So with using this application of this law, we are able to calculate the refractive index of different type of oils, or you can say the ghee or other ghritas in case of Ayurvedic drugs. As per example is also given of Brahmi Ghrita. As per IPI, the refractive index was calculated using 40 degrees Celsius and it is 1.454 to 1.465. So this is the refractive index of Brahmi Ghrita. So it is, yes, applicable in case of oils, Ghritas also. Next is the physical parameter, again optical activity, and we know there are different type of compounds also contain some complex substances like chiral carbon is also there. Suppose if a compound has 4 different functional group, so that carbon is known as the chiral carbon. So this is applicable in case of different type of, you can say, avleh which contains sugars. The sugars are optically active. So yes, we also know that the substances which can rotate the plane of polarized light either towards right side, they are known as dextrorotatory, and the substances which can rotate the plane of polarized light over the left side, they are known as the levorotatory. So with the utility of these substances of the chiral carbon, we are able to calculate the optical activity also. So there are different other parameters of standardization like pH, specific gravity, viscosity, solubility, melting point, disintegration, saponification value, acid value, peroxide value, iodine value. Yes, of course, in case of avleh, or you will say, not avleh, asav and arishtha. Asav and arishtha, in which alcohol is also present. We all know that self-generated alcohol is produced in case of our very famous Asavas and Arista. So alcohol content determination is essential and most important parameter in that particular formulation, and of course the density test for Gritas. So there are different parameters. So next, apart from the physical one, now we are going or move towards the chemical parameters. So chemical simply mean we are using some chemical test, or you can use the analytical methods like chromatography and spectroscopy for the evaluation of the crude drug or the herbal medicine or the herbal drugs. So in the chemical, or you can say the phytochemical screening, there are tests for different metabolites, like test for glycoside, test for steroids, test for saponins, test for alkaloids also. So let's just recall in a brief. Generally we do this topic in a brief. So test for, you can say, the steroids, there are different tests like Salkowski reaction, Lieberman test, and Lieberman-Bouchard reaction is also there. So if we talk about the Salkowski reaction, what we will do, we will prepare first of all an extract of the drug. Suppose you have to do this test on a drug. So what you will do, you will take a crude drug. What you will do, prepare an extract, then you will perform this test. So prepare few milligram of the residue of each of the extract that you have already prepared in 2 ml of chloroform and 2 ml of concentrated sulfuric acid, and again the test tube was shaken for few minutes. The development of the red color in the chloroform layer, basically over here I also mentioned the chloroform layer, if the red color will appear, it will indicate the presence of steroids in the sample. So in the same way, Lieberman test is there in which we are using the acetic anhydride, and Lieberman-Bouchard against advanced form is there, we are using other organic solvents and other chemicals. So these all identification tests are necessary, and these are the preliminary tests for determination of particular metabolite in a particular sample. So next one, alkaloid, there are different tests like Mayer reagent, Dragendorf reagent, Wagner's and Hager reagent. If we talk about the composition, so what is this Dragendorf reagent? It is potassium bismuth iodide solution. Mayer reagent is potassium mercuric iodide. Wagner is the iodine potassium iodide solution. Hager's solution is a saturated solution of picric acid, whereas the tannic reagent also used for determination of alkaloids in a particular herbal drug or a crude drug. So this is the prescribed procedure. Over it is mentioned. Next one is the chemical test for saponins. Their force formation test is very important there for determination of saponin glycoside, and also we'll do the using the blood sample hemolytic test is also there. So this is applicable for saponin containing drugs like licorice, shatavari. There are different type of drugs are which contain saponin glycosides. So yeah, this is a force formation test. Then the test for tannins, amla, beheda, hard, arjun, ashoka. These all are the tannin containing drugs, and we know tannins are substances. These are polyphenolic compounds which cause protein precipitation. So here is a representation again of ferric chloride test, which will turn your sample into black color. You can also do the lead acetate test, that is the gelatin solution test. Potassium dichromate test is also there for detection of tannins. Next is the determination of proteins. So we have ninhydrin test, biuret test, xanthoprotic test, and million reagent is also there. I am not going much detail into it because of time constraints. So this is the general test for proteins. So next one is the chemical test for carbohydrates. So generally for determination of carbohydrates, we are using two tests, Morris reagent and Barford reagent. And they are also different for sugar, or you can say the reducing and non-reducing sugar, we have separate test also. So in case of Morris reagent, we are using alpha naphthol and of course sulfuric acid also there. And in the end product, we have obtained this kind of, you can say the purple color is obtained, or the violet color is obtained in case of Morris test. Next one is the Barford reagent. Again, we are using copper acetate and 1% acetic acid solution. So red color precipitate will indicate the presence of carbohydrate in a particular sample. Next one is the test for flavonoids. So again, synodal test is also there, which will give pink color. And of course, the chemical reagents are magnesium turning is the most important, HCl plus ethanol are we are using. Next one is the, yes, of course, after the preliminary test, we have to use some sophisticated instruments for determination of our particular constituents in a herbal drug or a crude drug. So we are using chromatographic and spectroscopic approaches to do that. And of course, the father of chromatography, Michael Estes, the credit goes to this personality. In 1906, he invented the chromatographic approaches. So chromatography comprises of two words, chromatos and graphos. Chromatos mean to color and graphos mean to be written. So chromatography generally use two phases, mobile phase and stationary phase. As the name indicates, mobile phase will move and stationary phase will remain stationary. So based upon the affinity of the compounds which are present in our sample, either towards stationary phase or towards mobile phase, they are separated. Yes, so chromatography is a separation technique, it is a purification, it is a ratification, it is again the very important technique for, you can say, the visualization or separation, isolation of secondary metabolites or different constituents present in a particular sample. So next, there are different types of chromatography techniques like paper chromatography, TLC, HPTLC is there, gas chromato is there, ion exchange chromatography. So in this lecture, we are only just able to see the brief about different chromatography technique because we don't have much time to do that. So let's start the paper chromatography. Again, basic principle based on the principle of generally partitioning. So in this, we are using a specialized filter paper known as the Whatman filter paper of different grades. Again, it is of different types like radial, we can say the vertical, 1D, 2D, dimensional. There are different types of paper chromatography approaches are there. So here is a diagrammatic representation of this radial chromatography, and here is the paper chromatography, simply the vertical one. Here is the sample was applied with the help of capillary, and again this sample is a mixture of different phytoconstituents and we are able to see it. The separation will be achieved by using stationary phase and mobile phase. Next one is the TLC, again very important, thin layer chromatography. We generally use this technique in our laboratory, in analytic industry, in herbal drug industry, even in the allopathic synthetic chemistry also. So TLC again based upon the principle of adsorption. It has again two phases, stationary phase and mobile phase, and again separation is based upon the affinity of the compounds present in the sample either towards stationary phase or towards mobile phase. And generally in case of normal phase chromatography or normal phase TLC, we are using a polar stationary phase and non-polar mobile phase. Next one is HPLC, again very important technique used for qualitative as well as the quantitative estimation of our herbal drug extract or any other formulation. Yes, again it is of different type like reverse phase is also there and normal phase chromatography, HPLC is also there. Generally in industry we are using the reverse one. Next one is the difference between HPTLC, again high performance thin layer chromatography and thin layer chromatography. So these are the different representation comparison sheet of HPTLC and TLC. As you can see the layer thickness layer, in HPTLC we have generally use the thickness of 100 micrometer and in TLC it is 250 micrometer. Next is the efficacy. Yes, it has higher efficacy, it's less efficacious. Basically HPTLC is the most, we can say the more advanced form of TLC. In this we are using some sophisticated instrument and manual work is very less, but over here we have to do each and everything is manually. We will prepare our laboratory, we will prepare our TLC in our laboratory, we will sometimes also use the pre-coated one, but generally in our laboratories and colleges we are generally prepare our TLC plate by our own using adsorbent like silica gel G in water. So here is a manual spotting is there, auto sampler is there, and HPTLC again advanced form hai. Next, yes, this is our instrument setup. As you can see, applicator is there, developer is there, scanner, interface, and of course the derivatizer is there. Next one is the column chromatography, very important technique, again used for the isolation purpose. Separation and identification is also there. Again used, basic principle is adsorption. developed by DT Day in 1900 along with the Mikhail Estes, that is the father of chromatography. Gas chromatography is also there using GLC, that is gas liquid chromatography, and also GSC, that is gas solid chromatography. It is widely used for the determination of oils. Even in case of Ayurvedic oils, we are generally used for GC setup or the GC of different oils. Next, after chromatography, now we have spectroscopic techniques. So after isolation, we have to identify the structure of the compound. For the structural elucidation, we generally go for the spectroscopic techniques like UV, IR, NMR, and MAS, and each have different application. As you can see, in case of UV visible spectroscopy, we generally have some applications like it helps us to predict the N saturation, presence of different carbonyl groups, and also the molecule will multiple bonds show absorption at different wavelength. So it is basically Lambert-Beer law is used in which we can say absorption is directly, or you can say it is directly proportional to the molar absorption coefficient, that is the coefficient, and molar concentration plus optical path length. So this is a complex procedure. It is a very important technique for determination of qualitative as well as the quantitative estimation of herbal drugs. So next one is you can say the range is very important, 200 to 800 nanometer in UV visible spectroscopy, and for IR spectroscopy, yes, it is helpful in determination of different type of functional group present in the sample. Hydrogen bonding is there, aromatic region, also tell us about the geometric isomerism. Next one is the, yes, vibration is there, stretching, bending, vibration is there. It is a very detailed concept. FTIR using, nowadays we are generally using this instrument that is known as the FTIR in the laboratory also. Next one is the NMR, again carbon NMR and hydrogen NMR. It will tell us about the number of carbon atoms and hydrogen atom in our particular, you can say the extract or isolated compound, and there are different advantages also. Skeletal structure, so it helps in the determination of different structural elucidation as well as the aromatic region के बारे में पता चलता है. Next is the relative number of protons present in the molecule. So this is the representation of the NMR. And next one is the mass spectroscopy. Yes, molecular weight के बारे में हमें पता चलेगा. Mass spectroscopy से again very important technique for determination of molecular weight as well as the functional group. So next, after the physical chemical evaluation, now we are moving towards the biological. Bio means in the tissue, in the sample, in the animal. So generally biopsies are performed on animal or animal tissue, whole animal, isolated organs, and we generally test our herbal drug using these tissues. Yes, so toxicity studies are also a part of biological evaluation, in which we generally use for in vitro and in vivo toxicity studies by using different methods. Like for example, in vitro by using MTT assay, neutral red dye, ELISA, and cell lines. Whereas in vivo study we are using branch shrimp assay, subchronic and acute studies are also there. So here very important table is there of different Ayurvedic formulation. As you can see, this is the Ayurvedic formulation Arka, Asa Varishtha, Ableha, and Churna. So there are different tests over here. This is a test that are mandatory for these formulation. Say for example, for Aswa we have to calculate the description, color, odor, then pH is mandatory, volatile oil content is mandatory, specific gravity calculate करते हैं. Next, clarity test very important. Then test for heavy metal, again alcohol content determination भी आएगा. That is very important for Aswa and Arishtha, again very important test. This is written over here, alcohol content determination. And test for methanol, very important. So these are the common tests, like test for specific pathogen, test for pesticide residue, organochloride pesticides, pyrethroid, etc. So again for Avleha, as these Avleha are the sugar containing substances, we can take sugar, jaggery. So it has a standard test that is mandatory, that is the reducing sugar and total sugar. This is important for Avleha. Again Churnas के लिए test दी है. Microscopic is there, microscopic is also there, LOD, ash value, water soluble ash, total ash value. There are different types test like heavy metal toxicity, lead, cadmium, mercury. So over here also Gritas के test दिए हैं, Vatica, Gutica, and single plant material. So these tests are very important for any herbal drug standardization. तभी हम उसको किसी भी formulation में किसी भी तरह से हम यूज कर सकते हैं ताकि उसकी एफिकेसी को इंक्रीज किया जा सके. So over here these tests are given again for Gritas, Vatica, Gutica, and single plant material. So these are the references I have used to prepare this video. So you can watch all the lectures of chromatography spectroscopy from this channel, that is Knowledge Pharmacy. So in the end, I would like to thank you all for your patience and listening. Thank you for your valuable time.